Hello and welcome to the video on heredity. So we start this week with some definitions. First, heredity is the process of passing traits from biological parents to children. And then genetics is the study of heredity or inheritance. So those terms are often confused. Genetics is really the study of heredity. Alright, this shows us a picture of some DNA. It's wrapped up tightly in our cells in what are called chromosomes. Chromosomes are these long strands of tightly coiled DNA. Again, it has to be coiled up in order to fit inside of our cells. The segments of DNA called genes contain the traits that each person inherits. So within each chromosome there are several genes, and then the gene, remember, is going to have the information to make specific proteins. Next, we need to define autosomes and sex chromosomes. So this is a human karyotype. It shows all of your chromosomes. Obviously, this is under a microscope. But you can see most of them are autosomes. You get one from your mother, one from your father, and they pretty much match. And that's most of your chromosomes. Then we have one pair of sex chromosomes. So this right here is a male because it has one X and one Y. If there were two X's, it would be a female. And the sex chromosomes are going to determine your gender. So the autosomes are most chromosomes, and they're the same in males and females. Sex chromosomes are different in males and females. Alright, why do gametes contain only a single set of 23 chromosomes? So remember, gametes are the sex cells that are produced through meiosis. And the reason is because at fertilization, the chromosomes from the father contained in the sperm align with similar chromosomes from the mother contained in the egg to create a set of 23 pairs or 46 chromosomes. So that zygote that's formed has the correct number of chromosomes. And if it didn't have the correct number of chromosomes, it wouldn't be able to divide and develop. Alright, this figure shows how the gender of a child is determined, and in humans it's different in different species. Basically, you're going to get an X chromosome from your mother, no matter what. And from your father, you may get an X or a Y. And if you get two X's, you're a female. If you get an X and a Y, you're a male. So that's gender determination. Question for you. The genetic information in male offspring is... And the correct answer is A. It's inherited from the father and the mother. Alright, next we need to know what a locus is. So it's the location of a specific gene on a chromosome. So in this case, this light blue gene, that's its locus, and they have numbers on specific chromosomes. They'll tell you the chromosome number and then the location on the chromosome. An allele is an alternative form of a gene. So in this case, on the left here you can see there are two blue alleles, and on the right there's a blue and a brown allele. So this might be the example of eye color. And on the left we have someone that's homozygous, meaning they have similar alleles or identical alleles at a given locus. So this person has both alleles are blue, so they're going to have blue eyes. On the right, this person is heterozygous. That means they have different alleles at the locus, so they have one blue and one brown. Then we need to know what dominant and recessive alleles are. So dominant alleles are going to overshadow recessive alleles, and then recessive alleles are going to be overshadowed. So here on the left we have a heterozygous person, so they have one brown and one blue allele, but since brown is the dominant allele, this person has brown eyes. In the middle we have someone that's homozygous. They have the two dominant alleles, so they're going to have brown eyes. And on the right we have someone that is homozygous. They have the two blue alleles, they're going to have blue eyes. So in order to have a recessive trait expressed, you have to have both alleles of blue eyes. Codominant is when neither allele can mask the expression of the other allele. So an example of this in humans is the ABO blood type. So for example, if you get AA, you're going to be blood type A. If you get AO, you're also going to be blood type A. But if you might be AB, so A and B are codominant, so you can be both. This is an example from plants. So some of these flowers from one plant are going to be red, and then some are going to be white. So the red is denoted with the genotype capital R, and the white is little r. But if you mix them together, you'll get pink flowers. So some flowers, it's either, if it was a dominant recessive relationship, you would get either red flowers or white flowers. But since they're codominant, you can get pink. Then next we need to know what a genome is. So a complete set of genetic information for one person, which is carried on the 23 pairs of chromosomes, is called a genome. Next, we need to describe polygenic inheritance, which is the phenomenon whereby genes at two or more loci contribute to the expression of a single trait. So in the example we show with eyes, we show there's one location where you have either brown or blue. In reality, it's more complex than that, and most genes are more complex than that. Maybe in plants it may be simpler, but in humans most things are polygenic. So you may have heard that baldness comes from your mother's father. Well, now in reality we know it's much more complex than that. Alright, what is the location of a gene called? And the correct answer is locus. Sounds kind of like location. Alright, next we need to know what genotype and phenotype are. So genotype is the genetic information stored at the locus of a gene, even if those traits are not expressed. So, for example, the genotype here for red is capital R, and then the phenotype is red. So the phenotype is the detectable outward manifestation of a genotype. So the phenotype of the flowers is red. Phenotypes may be things like eye color or height or skin color or really most things that you can think of that you can observe. All right, next we're going to look at sex-linked inheritance. So say we have a trait, say it's color blindness, and that's one of the most common ones. It's carried on the X chromosome. So this female here has one recessive allele and one dominant allele. So if she inherits the allele for this condition, the allele on her other X chromosome would overpower the recessive allele, so she would be a carrier, and she would not have color blindness. If we look on the right, if the man gets this recessive allele, he only has one X chromosome, so the trait would be expressed in him, and he would be colorblind. So sex-linked inheritance is going to be an allele or a gene that's on one of the X chromosomes generally. All right, DNA mutation. A permanent change in genetic material is known as a mutation. Some mutations occur spontaneously, just due to errors in copying DNA, but that's pretty rare. They can result from exposure to radiation, certain chemicals, or viruses. So, for example, that's why cigarette smoking causes cancer, because it can cause mutations in the DNA. All right, genetic disorders can be single gene disorders or chromosomal abnormalities. We're going to look at for some single gene disorders and look at autosomal dominant inheritance. So in this case, we have an affected father who has the disorder and an unaffected mother. So in this case, he's heterozygous. He only has one dominant allele, but that's enough to give him the disease. And the mother is not affected. And if we look at their children, their children have a 50% chance of developing the disorder. It's autosomal dominant. So an example of that would be Huntington's disease. And if we look at someone who's autosomal recessive, in this case both parents are heterozygous, so they have the normal allele for the normal gene, and then they have the defective recessive allele. So in this case, they're going to have, statistically, they're going to have 25% chance of having an unaffected child, a 50% chance of having a carrier who's not affected, and a 25% chance of developing the disorder. So the chances of developing the disorder are less because you have to get the recessive gene from both parents, as opposed to autosomal dominant. All right, which statement about sex-linked inheritance is true? And the one that's true is, let's see, almost all these traits are carried on the X chromosome. All right, next we're going to talk about cystic fibrosis. It's caused by a defective gene on chromosome 7. The protein produced by this gene normally regulates the transfer of sodium into and out of cells. But because of the mutation, the transfer is impaired, and when the transfer of sodium is impaired, the transfer of water normally follows sodium. This causes exocrine cells to secrete profuse amounts of thick, sticky mucus. The excess mucus causes particular problems in the lungs and intestines, where it leads to complications such as infections, especially in the lungs. That thick, sticky mucus is harder to get out. And in the intestines, it can lead to also infections, blockages, or difficulty digesting food. And they have a lowered life expectancy. It's rare because it is an autosomal recessive disorder. So chromosome abnormalities result when large segments of a chromosome or an entire chromosome are missing, duplicated, or altered. The most common disorder is nondisjunction. So this first figure shows normal disjunction, where as you're going through meiosis, the chromosomes separate, and you get cells with half the number of chromosomes, and then when fertilization happens, they have a complete set again. This figure shows nondisjunction, so those chromosomes, when they fail to separate during meiosis, you get one cell. So at fertilization, the cell on the left is going to have one chromosome, and the cell on the right is going to have 3 chromosomes. So nondisjunction is when a pair of chromosomes fails to separate. So one daughter cell ends up with 0 chromosomes for that pair, and one ends up with 2 chromosomes for that pair. And this is only showing 1 chromosome, so normally they'd have, like the cell on the left would have, say, 22 normal pairs, 22 single chromosomes and 1 missing, and the one on the right would have also 22 single chromosomes, and then this one where they have a double chromosome. And that can lead to problems later on with cell division. So Down syndrome is caused by an extra copy of chromosome 21, so some nondisjunction probably happened. Some of the symptoms include a low muscle tone, a single deep crease across the palm of the hand, slightly flattened facial profile, upward slant to the eyes, cognitive delays, intellectual disability. It's associated with a lot of birth defects, including abnormalities of the heart and kidneys. And the reason it's the most common, if we go back to our karyotype, we can see chromosome 21 is the smallest chromosome, so it's easier to survive with an extra there. If you have an extra of chromosome 1, for example, that embryo as it's developing isn't going to be able to divide, and it's not going to be able to successfully divide. So that's Down syndrome. With non-disjunction, it can happen with sex chromosomes. And if it happens with sex chromosomes, so on the left here we have one cell has two X chromosomes, and then when it's fertilized with a Y, the genotype is XXY, and that is Klinefelter syndrome. Klinefelter syndrome is caused by an extra X chromosome in a boy, and these symptoms include undeveloped testes, sparse body hair, unusually long arms and legs, enlarged breasts, low intelligence, and sterility. And then if we look on the right, this cell got no X chromosomes, and if it was fertilized with an X chromosome, that's called an XO genotype, and it results in what's called Turner syndrome, when a girl is born with only one X chromosome. Some of the symptoms are the secondary sex characteristics fail to appear. Also, they'll have a webbed neck and a short stature. All right, next we're going to look at a Punnet Square. We use these in genetics to determine the probability that different offspring will have different traits. So in this case, we're looking at the offspring of a man who is homozygous dominant for brown eyes. So his genotype is capital B, capital B. And I added color here, but you don't have to. And then a woman who's homozygous recessive for blue eyes, so her genotype is little b, little b. So what we would do is we'll put the man on top, and it doesn't matter if it's the man or the woman on top, and the other one's on the side. So I put the capital B's on the top from the man and the lowercase b's on the side, and then I'm going to bring down those capital B's and bring over those lowercase b's, and then I'm going to bring down the next capital B and bring over the next lowercase b. And so then each box is going to represent the chances of having that type of offspring in that type of genotype in their offspring. And in this case, they're all the same. But if you're doing a Punnet Square for someone who's heterozygous, you would get different combinations in there. So in this case, the genotype is 100% capital B, lowercase b, and the phenotype is going to be 100% brown eyes, since brown is dominant to blue. All right, next we're going to talk a little bit about evolution. So we need to know what a theory is. It's a well-substantiated explanation of some aspect of the natural world based on a body of facts that have been repeatedly confirmed through observation and experiment. These are reliable accounts of the real world, not guesses. One reason it's a theory is because we can't prove it. If we can prove something, it's a law, such as gravity. Gravity is a law. If you don't believe that, try making your phone float. See what happens. But a theory is something we're pretty sure it's correct, and we have lots of evidence to support it, but we can't prove it. So the theory of evolution, as simple as I can make it, biological evolution is descent with modification. So as organisms change, like you're different from your parents a little bit, and these changes in gene frequency in a population from one generation to the next, that's called small-scale evolution, and the descent of different species from a common ancestor over many generations, that's large-scale evolution. Evolution can help us understand the history of life. So you may hear it described as survival of the fittest, which is true if a species, if some small change happens that makes something more fit, better at reproducing or better at surviving, those traits are going to be favored, and they're going to be passed on to subsequent generations more frequently than other traits. So the theory of common descent. The central idea of biological evolution is that all life on Earth shares a common ancestor, just as you and your cousins share a common grandmother. Through the process of descent with modification, the common ancestor of life on Earth gave rise to the fantastic diversity that we see documented in the fossil record and around us today. So for example, we hear that humans come from apes, but we actually share a common ancestor that was neither human nor ape. Humans evolved from one branch and apes from another. The process of natural selection and how it leads to genetic changes in a population of organisms. And I don't think this is real, but it explains it well. So these birds say young green beetles are favorite. So they're eating all green beetles, maybe because they taste better, although birds eat some pretty gross stuff, so maybe it's just because they're easier to see. And so you can see at first there's mostly green beetles in the population. After many, many generations, you have roughly equal numbers of green and brown beetles. And then after many, many more generations, all the green beetles have been eaten, and the only ones that are surviving are the brown beetles. So that's natural selection or survival of the fittest. And next we're going to look at some evidence that supports the theory of evolution. So there are lots of evidence. So one thing is fossils. Fossils document species that are related to other species, and we can look back at fossils and figure out how old they are and see how they're related. Biogeographical evidence is looked at to show where species are and what unique features they have. So depending on the environment you live in, you adapt to that environment. Molecular evidence looks at biology and the species DNA or genetic code to show shared traits and relations between species. Now with DNA sequencing that we have, we can compare two species' DNA and see how related they are by how similar their DNA is. Direct observations are used to see smaller scale evolutionary processes in some species. We can see this, for example, in bacteria. We can look at how antibiotic resistance develops in bacteria in just a few generations, and we can see this in a matter of days in something like bacteria that reproduces really quickly. And then anatomical evidence is used to compare physical features and figure out when they developed. So for example, if you look at the flipper bones in a whale and the arm bones in a human, they're actually pretty similar if you look just at the bones. And you can find other evidence that supports the theory of evolution as well. Last thing I want to talk about is multifactorial inheritance. So some diseases are influenced by both genetics and environmental factors. Environmental factors influence disease progression or whether a disease even develops. And the human microbiome also plays a role in regulating genetic expression.